Welcome to Winchester Cafe Sci Online, and a special welcome to those joining us for the first time. A recording of this talk will be posted on our YouTube channel, where you'll find recordings of all our online talks. I will put details in the chat. It would help us reach a wider audience if you could subscribe to our channel. You can also find the links on our website, wincafesci.org.uk. If you enjoyed tonight's talk and you're not on our mailing list, you can sign up for it there to be notified about future talks. After tonight's talk, we'll take a break for a few minutes before the Q&A, so please put your questions into the chat and we'll read them out. That includes our viewers on YouTube. Tonight's speaker is lecturer in Evolution and Palaeobiology in the School of Biological Sciences at the University of Southampton. Over the last year, he and his students and colleagues have had to adapt to the global situation, but science hasn't stopped and neither has he. A new dinosaur or two and a new understanding of mammalian physiology at the group's appearance are just two of the things he will discuss in his talk. When he's not working on dinosaurs or STEM mammals, he's the program leader for the Zoology degree program and the MRes Evolution from the Galapagos to the 21st century. This exciting program takes students to the Galapagos and explores evolutionary processes across 9 schools at the University of Southampton, from biology to engineering, medicine to philosophy. Evolution underpins the science of biology. Please welcome Dr. Neil Gosling.
Give me just a second. It's lovely to see you. Thank you ever so much for having me. I'm going to talk this evening about 3 projects that I've been involved in over the last, well, it's the last several years, but that have come to fruition and have been published this year during the time of COVID. So what do I do? I'm a very broadly trained evolutionary biologist. My undergraduate degree was in botany and zoology at the University of Reading. And then I stayed at Reading and I did my PhD in evolutionary developmental biology. So looking at how patterns of development are shared across the animals. You can do it in plants as well, but I was looking at animals and looking for common pathways, common developmental mechanisms and patterns, which highlighted evolution. And that led on to working on fossilized embryos. I've always wanted to be a paleontologist. That's you know, fossils are the fun bit, but I didn't do geology. And it was when I started doing my degree, it was still pretty much the case that it was, if you wanted to be a paleontologist, you studied geology. Now it's pretty much half and half. There are biologists and there are and geologists, and both of them sort of come together from a slightly different perspective and end up doing the same thing. So I was working on fossil embryos in Bristol and it was phenomenal. Had a lovely time. And I've sort of diversified into broader questions of evolution and paleo as I've gone through. And I'm going to tell you about a couple of those tonight. When I was, the thing that really got me wanting to be a paleontologist was in the mid-90s, when it was really shown with the beautiful fossils that were coming out of China that birds are dinosaurs. I've got Archaeopteryx up on the wall there, and I have the most beautiful reproduction of Archaeopteryx sitting underneath it. Just remarkable. Birds are cool as it is, but I don't know, there's just something about the fact that dinosaurs didn't go extinct 66 million years ago. That's fun. And I get to have fun. I'm very, very lucky. So let's just, there we go. So tonight I'm going to talk to you, it sounds a bit technical, but I'm going to hopefully introduce you to some exciting things and the way that paleontology isn't a paleobiology. The difference is it's a very sort of nuanced difference. A paleobiologist is thinking about the organisms as how they would have lived, how they interacted with their ecosystem, thinking about extinct organisms in exactly the same way as you would think about living animals now. So that's the difference between a paleobiologist and a paleontologist. if you call me one or other, it doesn't really matter. But what I want to show you is that actually paleo has become really high tech, and we're using all sorts of interdisciplinary approaches that allow us to really understand the fossils that we're looking at and to get all of the information we possibly can out of fossils. So I'm going to talk to you about why 3D is better than 2D for looking at fossils. I'm going to talk to you about a project that a PhD student called Katie Williams, she's now Dr. Katie Williams, she worked on a phenomenal project to understand the structure of bone in living birds, with the hope that we'll be able to apply that exact same information, exact same technique, and get the information out of fossil birds, and I'll tell you why that's important. Then I'm going to introduce you to Vectera venator inopinatus, which sounds like a mouthful, but we had a focus group. We spoke to all the 5- and 6-year-olds we could find. They could all pronounce it, so it was perfect for a dinosaur name. And then we're going to look at Morganeucodon and Cuniotherium and the age that they got to. These are stem mammals. They're very early mammals. And I'm going to give you I'm going to take you on a vivid journey through the biology of the earliest mammals. And I know that you're all thinking this at the moment. They're the little ones which were creeping around at midnight, eating what they could, the scraps that fell from the tables of dinosaurs. Dinosaurs didn't have tables, but it's a vivid word picture. Wandering around, hiding in their footprints, getting out of the way. And I'm going to show you how that idea has changed and talk to you about what we know about the evolution of the earliest mammals. So why is 2D not as good as 3D when we're looking at fossils? Well, if we go to this paper here, Chen et al. 2004, small bilaterian fossils from 55 million years before the Cambium. This is why 2D is not as good as 3D if we can work on it. These are thin sections, okay? We've got a piece of rock has been taken, it's been ground down, it's been stuck onto a microscope slide, ground more and more and more until it's thin enough that light can pass through it. And then we get these wonderful structures, and we can see these little spherical structures. It's about 0.5 millimeter to 1 millimeter in diameter. And what we see here, and this is remarkable because this is 55 million years before the Cambrian. The Cambrian is when bilaterally symmetrical organisms, animals, appear in the fossil record. And what we're seeing here, if we're to believe this, is an animal, or a fossil rather. We've got a mouth, we've got a gut, we've got body cavities, coeloms, we've got sort of sensory pits around the outside. Here's another section through another one. Sensory pits are really obvious here. You can see the gut. And we can travel through and we can look at this organism, and we can, this is remarkable. I mean, we don't have fossils of animals really this sort of distance back. We now understand that we do have some, but not these little things 55 million years before the Cambrian explosion, well, before the Cambrian, let alone the Cambrian explosion. So this is really remarkable. But if you've got a remarkable statement, you have to have sort of pretty remarkable evidence to back it up. And you can put all these sections together and you can get this beautiful, funny little sort of bilaterally symmetrical mouth at that end, sensory pits here, crawling along the surface of Cambrian's oceans, Vern animalcula, the dawn animal. It's a nice name, isn't it? Sounds good. The trouble is, when you're using 2D slide, when you're looking at something which is about 1 millimeter in diameter and you grind it down, you're lucky if you get a single slice out of the thing that you're interested in looking at. And it's destructive, it's gone. And so what's been done is lots and lots of pieces of rock have been taken, lots and lots of thin sections have been taken, and they've built it up and they've sort of tessellated everything until they've got this wonderful organism. The trouble is it's not really accurate. If you take it for a moment, just to give you an idea of the period of time that we're talking about, I mean, if the Earth forms in well, we'll go to Big Ben. Big Ben bonged for the first time in 1859. So that's a jolly good time to start any story and any place, because that's when Darwin published The Origin of Species. And we take a 125-kilometer journey, giving it away, 125-kilometer journey from the start of the Earth to the present day. So we're changing time into a linear journey, so we get a better idea of the length of it. Because frankly, if you take it as a clock, humans appear in the very last second of the last hour of the 24 hours of it. It's too abstract a concept to think of time using a clock. But if you use a journey, then that makes better sense. And what we're looking at here is if you come down all the way and you get to Winchester. Winchester is about the point at the base of the Cambrian. Okay, Winchester is where animals appear in the fossil record. On that same journey, life begins in Slough. You can make of that what you will. But we're talking, this idea that an animalcula is real and is a bilateral, it suggests it's starting off before the Cambrian, and that's simply, it's not really accurate. Okay. And actually, if you take these beautiful little fossils and you remove them from the rock, so they're entire 3D specimens, and you take them to a synchrotron or you do X-ray scanning, so you're now not grinding them and hoping for a single segment, section. What you're doing is you're using high energy, high resolution CT scanning, and so from something which is approximately a millimeter in diameter, we can get out several thousand slices now. So we can then reconstruct this on the computer. And what you can see here is actually, if you look at these, an individual section in isolation, well, there's a mouth, here's a through gut, here was a couple of body cavities, coelomes, and there we go. There is our bilaterally symmetric. You can look at this one as well. It's exactly. So we're not seeing the real representation of what these artifacts, fossil things are in life. We're seeing an artifact of 2D preparation. And that's really setting up the whole story for where we go from here. From 2005, when I first started, this was the work that I was doing in 2005. This work was published in 2012 by collaborators and my former lab mates, John Cunningham and Phil Donoghue was my boss. Stephen Bankson I collaborated with as well. I mean, these guys are the group which really started off using high resolution CT scanning to develop 3-dimensional models of what we're looking at in the fossil record, and taking separating geology 500 million years in the ground and looking at what biology is preserved. Biology is really important. A lot of the time on the insides of these surfaces, you're not preserving biology. What you're preserving is little tiny crystallization events. And so you've got these little tiny, almost geodal infillings, all these dozens of lines. These are little areas where calcium phosphate, in this case, has started to come out and crystallize on a surface. And that surface may originally have been a biological structure, but it's completely overgrown with geology. So working out what we're looking at is really important. So there we go, straight off, looking at something in 3 dimensions is much more useful and much more informative than looking at something in 2 dimensions. So how does that bring me on to Archaeopteryx? Well, one of the issues is, this is the fossil that got me interested in all of this. Archaeopteryx was the name initially given to a feather that was found in 1861. In 1859, when Darwin was writing The Origin of Species, he is really at a loss, a bit confused as to why we don't have lots of transitional forms linking the great clades, the great classes of different organisms. And what he argued was that it's there, we just haven't explored the fossil record enough, and the fossil record that we have is fairly incomplete. So we would find these things, but we just haven't looked hard enough yet. And if in the places where we've seen it, you know, the fossil record isn't perfect. 1863, this body fossil turns up in a limestone quarry, in Solnhofen in Germany. And it was remarkable. We have these feathers, animals that have feathers are birds. And that was one of the reasons why, I mean, a weird bird, it's got this long bony tail, its brain case is over here. And when you find the jaw is in here as well somewhere. The Berlin specimen is prettier, but you can see all these teeth. Birds don't have teeth. They're pretty rare, especially in hens. But you've got these feathers, got this long bony tail, there's fingers as well with claws on. It's clearly quite a weird bird, but it's got feathers, and the only animals that have feathers in 1863 are birds. So when Richard Owen, who was not an evolutionary biologist of any way, shape or form, bought it for the Natural History Museum at a price of £700, which by today's money is a phenomenal. We just don't have that kind of money to be spending on fossils. No public institution has that. It went into the collection, went in as a bird, and that was that. Thomas Henry Huxley, Darwin's bulldog, recognised that it was a transitional form, and right from the earliest point that we have it, and we're talking about evolution, birds were dinosaurs. We get the proof of this in 1992, 93, with the discovery of the beautiful specimens from China. But the problem is with Archaeopteryx is we've got the Solnhofen specimen here, which is frankly enormous, the Berlin specimen, which is the most beautiful, the London specimen, which is the first, and the Eichstätt specimen, which is really tiny. The mass estimates for this animal is 1/7 the mass of this animal. Are they even the same species? Are they even the same genus? They're all of these birds, these fossils, come out of pretty much the same deposit. So we have this specimen here, the Solnhofen specimen, which is 7 times larger mass-wise than the Eichstätt specimen. So are we looking at the same animal? Are we at the same species? Are we looking at the same genus? And so what we started to do, really the only thing you have to work with in fossils most of the time, despite the fact I'm telling you that, I mean, these fossils, feathers are absolutely beautiful, but how informative are the feathers as they are for telling you if it's the same species or getting a lot of biological information out of it? What we have is the bones, and we have bones of all kinds of different fossils. And so these are the things we're interested in. And you can take bones to places like SLS, the Swiss light source, which is not far from Zurich. You can go inside. Here is the accelerator running around. I love this picture. These are all the little hutches where you've got this ring accelerating particles to near the speed of light. In fact, they're going this way. Sorry, and then you've got these straight lines, straight tubes that take off X-rays, and that's where it's useful. So in these hutches, there's all kinds of experimental equipment that allow you to scan material at exceptionally high resolution. We're talking now about sort of 0.3 of a micron. Okay, there's 1,000 microns in a millimeter. We're looking at 0.3 of a micron. 1,000,000 microns in a meter. So you can get some really high resolution stuff. You can see the phases of deposition of minerals. So you can see that you've got this phosphate encrusting the inside of these little embryos. We can also take specimens to the MUVIS facility at Southampton. This is the largest of our scanners at the moment. The diameter here is approximately 1.5 meters. We can get, if we wanted to, we could scan a cylinder that was just shy of about 180 centimeters tall and about one meter in diameter. So we can scan very large things. In that case, down to a resolution of about 1/10 of a millimeter. If we've got smaller structures, smaller things that we want to scan, we can get them much, much closer to the emitter. So electrons come zooming out of here through our sample, which is mounted on this stage, and it's detected by this detector, which can move up and down. So you can take multiple pictures, and so you can get your 1.8 meters tall and your 1 meter in diameter. Remarkable, and we can get some absolutely beautiful stuff out of this. So here are some two-dimensional slices. These are normal histological sections taken through birds' humerus. And these are the equivalents. So we've got humerus here, we've got a leg here. You've got the same structure, the same section using CT scanning. So we've got our single slice, and here we've got our multiple stacked set of images. And our stacked set of images allow us to not just look and see what these canals are doing, but we can see if we make a 3D image. Here we go, here's some more. These are different ages as well. So very young, slightly older, up to about 18 months old. This is 3 weeks and 6 months, I believe. And we wrote this paper, and Katie went through and skeletonized the whole of followed all of those little pores through, all of those little blood vessels, all of those little lacunae, all the way through. Because what we can start to do, if you just look at a single slice, you're seeing the holes, but you can't tell the orientation of them. And the orientation changes with loading. So as the bird starts to fly, as the bird starts to walk, the way that the bone is put together, the way that it's modeled will change. And so you can tell the stage, the age of the animal in its development. And what we can do is, by looking at living birds at known time points, at known loadings and what have you, we can start to understand how bone changes. Because we've got all these bones in the fossil record, we've got Archaeopteryx, we've got 13 specimens, no one's looked. And so what we need to do is have an understanding of how bone remodeling happens through life. And we've looked at ducks, we've looked at pheasants, we've looked at all sorts of different things. Then we've got different time points through starlings and all sorts of different birds, pigeons, you name it. One of the things Southampton has is a beautiful zoarchaeology collection. So lots and lots of birds that have been collected from different archaeological sites, but also which have been opportunistically collected when someone has handed in a dead specimen. It's been aged, it's been looked at, and it's been prepped and kept in the collection for exactly this sort of thing. And what we can do is we can start to see the orientation, because if I just go back for a second, with this, we don't know if we've got an ovoid shaped tube, or if we're looking at a round tube. So if you take a drinking straw and you tilt it and then take a horizontal cut, you're going to get an elliptical shape, as opposed to the perfect circle if you cut it perpendicular to the length. So we don't know that. But if we put things together, if we start to look in 3 dimensions, we can start to understand the shape and the structure of blood vessels. We can start to work out the way that they change through development, and we can start, we haven't yet, but it's the next stage, we can go to the fossils, we can CT scan those, so with non-destructive testing, and we can start to age. Now, getting our hands on all the Archaeopteryxes is going to be a challenge, but that will ultimately allow us to really get down to the idea of whether or not we're looking at one species. Now, Archaeopteryx is just one example. There are lots and lots of Mesozoic birds. There are dozens of Mesozoic birds. Many look very, very similar. What we can do, hopefully, is to look and see if we have got multiple different taxa, different species, or if we're looking at different life stages of fewer species. And that might seem boring. Oh dear, we have this beautiful diversity. Oh, we've reduced it. Or not. And if we reduce it, that's actually just giving us a more accurate idea about the diversity of birds in the Mesozoic. if it turns out they're all adults, then we know that the diversity is as awesome as we think it is. So it's about getting the accuracy in the fossil record. So next project, and this is the one I'm sure many people want to hear about, a highly pneumatic Middle Cretaceous theropod from the British Lower Greensand. Chris Barker is an absolutely super PhD student. I'm extremely lucky to be working with him. Incidentally, these numbers at the bottom, the DOI numbers, are links that will take you directly to the papers, and they are all open access. So if you copy the DOI number that you'll see at the bottom into a URL, into a web address, it will take you directly to that paper, and you'll be able to read it straight away. So here we are, we're in Shanklin. The Isle of Wight is absolutely beautiful. It's one of the best places. It's one of the best places in Europe. It's in the top 5 or top 10 places in the world for finding dinosaur bones and finding dinosaurs. And as you come across from Southampton into Cowes, and literally across the middle, there's the fault and the unconformity that moves you from the Eocene into the Mesozoic. And we're in the Mesozoic. So up at the top, no dinosaurs. South of the island, lots of dinosaurs. And the beautiful thing about the Isle of Wight, depending on how you're looking at it, obviously if you have a house there, not so good. If you're a paleontologist, awesome, because it's eroding at about 1 meter a year, which means the cliff is falling down. It means that bones are being exposed. The whole of the UK is fantastic for fossils, but people do careless things like build shopping centres and towns on top of the land. And as soon as Tesco's really don't like you digging up their car park because you think that there might be some good fossils underneath it. I haven't tried it, but I've got my suspicions that they wouldn't be cool with it. But where you've got coastal erosion, if you've got the right kind of rocks, chances are you can find some good fossils. And we've got Sandown here. Sandown is brilliant for 2 things. Number one, there's the Dinosaur Isle Museum, which is phenomenal and has holotypes, original, unique specimens of taxa, of species. And then it was also the place where Darwin put pen to paper to start writing The Origin of Species. A little bit down the way is Shanklin. And what's odd about Shanklin is, well, first of all, let me just jump over. Let's come over to this side. On the southwest side of the island, loads of dinosaurs. You can find the dinosaur footprints that have washed out the cliff of the big ornithopods. You've got these wonderful, there's a sauropod found on a dinosaur farm. There's all sorts of wonderful material. And this region here is about 125 million years old and represents a phenomenal terrestrial deposit. Dinosaurs are terrestrial. Dinosaurs don't live in water. If you've got an aquatic marine reptile from the Mesozoic, it's not a dinosaur. So that's the thing that's a bit weird, is because here, Shanklin is about 116 million years old. It's about 10 million years younger. And it is a marine deposit. So loads of ammonites, there's ichthyosaurs, there's all sorts of other material that you can find there, which is clearly marine in origin. At that point, about 116 million years ago, there'd been a huge inward sea ingression over most of Europe. And so you just have these sort of little islands popping their heads up out of the water. And it's not the best place to find dinosaurs. So it was quite a shock when on Horse Ledge, underneath Knott Cliff, in the ferruginous sands, bones started to be discovered. And we were very lucky in that 3 amateur collectors, I don't like the word amateur collector, they do more collecting than I do. These guys just don't happen to get paid for doing paleontology. They found 4 bones and handed them to Dinosaur Isle Museum, and when they handed them to people at the museum, they knew that they were something special. Here we go. We've got this cervical. We've got 2 dorsal vertebrae from the thoracic region, and we've got this caudal vert. Since we discovered this and we went on the news and talked about it, it's amazing. I've worked on all kinds of exciting things like fossil embryos from the dawn of animals, 4 dinosaur vertebrae, and everyone goes mad, and I get onto BBC News 24. But that was really good because people then started to contact us and say, oh, we've found some material as well. So we have got a couple more bones. And if anyone is over in Shanklin and they find material that looks kind of bony, please take it along to the to Dinosaur Isle so we can start to build up more of a picture of what this animal is like. This is where Chris comes in. Chris is a really excellent PhD student. He's a really excellent dinosaur researcher. And he looked at this material and was talking to people at the museum, and they thought it was something new and unusual. And he teased all of this apart and spent an awfully long time. He's really thorough. He went through and looked at every single aspect of 4 vertebrae and realized that you couldn't put them into any existing taxon, any existing species. This was something new. So he carried on looking, because not saying that something isn't something else is one thing. Saying that it is something and being able to diagnose and say this is something new is a next step. And this is what Chris did. He spent an awfully long time going through, finding the characters, comparing it with every single other similar structure, other similar vertebra from other species, and started, I mean, here we go. These are all his beautiful diagrams and beautiful reconstructions and beautiful annotations, okay? Again, this paper is available from Papers in Paleontology. It's the very final paper in the printed run of Papers in Paleontology, which has now gone online. So I'm quite pleased because it's the last paper that's ever physically published, physically printed. But he went through, here's our caudal vert. Here's one of our thoracic vertebrae. This is beautiful. This was discovered and handed in by a chap called Robin Ward, who was over on the island celebrating his daughter's 11th birthday. Absolutely phenomenal find, beautiful fossils. Here we go. Here's another one. This was found by a gentleman by the name of Farrell. And the caudal was found by a gentleman, Mr. Lockyer. Here's another vert. This was also found by Robin Ward. Absolutely beautiful preservation, beautiful fossils. And what we have found is that it doesn't sit with any other known species. It isn't a sister taxon to any specific other species. It sits outside. It nests in all kinds of different places. Now, we have very few bones. And so we can't get an absolute firm diagnosis of exactly what it is. What we can say is it's a theropod. It's one of the cool dinosaurs that was eating other dinosaurs. But exactly what it is, we're not sure. Here's one of the trees that we built up, and it sits beautifully with tyrannosaurs and megaraptors. So it's exciting. This is just one of the trees that came out. It is not very well supported. So I am not saying that it is a Tyrannoraptor. I am not saying that. But the more we can find, the more material that we can find, the better we'll be able to pull it together and say exactly what it is. At the moment, it's a theropod. And here is an absolutely stunning, I love this picture. This picture hangs in my front room. This picture hangs in the lift lobby in biological sciences. This is by Trudy Wilson, and it is our representation of Vectura veneta's final journey. I said that Vectura veneta, that Shanklin, the deposit, was a marine deposit. What we think happened was that the land was maybe 5 or 10 kilometers, a little island 5 or 10 kilometers north of Shanklin. It had a bad day, it died, it fell into a river, it washed out to sea, traveled over to Shanklin, sank into the sediments, was quickly buried, because we don't, and we think that it's a single individual because we don't have any, we haven't got we haven't got more than one of each vert. So we think it was a single individual that was buried quite quickly, because we have vertebrae from along the spine. If they'd just been all dotted over, who knows? We could have said that it was broken up, but we think because we've got bits of from along the body, it hasn't broken up. It hasn't been fallen into a highly energetic sediment or water. It's traveled along, sunk down, and away we go. And I absolutely love the ichthyosaurs lurking in the gloom. I really like the ammonites using the body as a sort of a raft, in the same way that fish will seek protection underneath vegetation and other bits of decaying material that have washed into the sea today. Really beautiful example of art meeting paleontology and science. Which brings me to the third of my stories, and this is really exciting. Ellis Newham, again, another PhD student who I have been extremely fortunate to work with and supervise. Ellis is an amazing student. He's currently working in Bristol. He's a name to watch out for. We have this idea that the earliest mammals, so those of about 200 million years ago, early Jurassic, in fact, pretty much all the way through to the extinction of the dinosaurs 66 million years ago at the end Cretaceous, that mammals were small and that mammals were lurking, keeping out the way of the dinosaurs, snatching little bits of food, were generalists, snatching food wherever they could, not really making much of a fuss about their existence, keeping out of the limelight. Well, number one, we know that, as we've looked through the fossil record now, we've found some very much larger animals. We're talking about, things the size of beavers, things the size of raccoons, not tiny mammals at all. But we have got a group of animals, stem mammals, mammalia forms, the first group, the first individuals that you can slot directly into the tree of mammals. They're extinct, so they're on this. If you imagine that a phylogenetic tree, a family tree, all of the tips of your branches are alive today. They've got little leafy sprouts on the top, but some of them down at the bottom don't make it up to the crown. They're not alive now, and they're on the stem of a branch. That's exactly it. So we've got these stem mammals. They're the group, they're on our lineage, but they just don't make it into the sunshine, if you like. What we did was we CT scanned things again. And Ellis did a phenomenal job. Here we go. This is Morganucodon. This is Cuneiform. Not generalists either. In 2013, I was very lucky to have been working with Pam Gill, and we CT scanned the teeth. We also laser scanned the teeth of both Cuneiform and Morganucodon. And what we could see is every time you eat something, it leaves marks on your teeth, micro wear, little tiny scratches. Doesn't matter what it is you eat, you get left with abrasions and marks on your teeth. And in 2013-14, we had, from 2006 actually it started, all the way through to 2013, we had CT scanned and laser scanned the surfaces of these teeth from the earliest mammals. And we compared them with the teeth of living, recently dead, but extant mammals, where the group is still alive, things like bats, things like rodents. And compared, so knowing what living groups eat, we then compared the teeth of Morganucodon, this grey one, and Cuneiform, this little brown one, with the teeth of living animals, and compared the microwear. And we showed that the earliest mammals weren't generalists, they were specialists. Morganucodon ate beetles. Cuneiform ate soft-bodied animals, soft-bodied inverts like scorpion flies. So they're already specialist in their feeding. So that's good. We've ticked that off. We've now shown that mammals are not as, they're more interesting earlier on than we've given them credit for. We also know that we've got larger ones as the Mesozoic progresses. But what can we tell? What more can we tell about their biology? We know that mammals are derived from a reptile group, the mammal-like reptiles, the synapsids, which went extinct 252 million years ago in almost all lineages, except the one which leads to modern day mammals as we have. If they'd all gone extinct, we wouldn't be here. What more can we tell? When did mammals become furry? When did mammals become warm-blooded? When did mammals become active? When were they, running around? What were they doing? What can we tell? Well, the trouble is, although there are some beautiful specimens, and we've got isolated long bones, we've got a beautiful skull of Morganeucodon, we've got a couple of them, but we haven't got very many. The thing that really preserves beautifully in the fossil record are teeth. I mean, Morganeucodon actually gets its name. Don means, don't, tooth. Morganeucodon is the tooth of Glamorgan, and it's found because lots and lots of material was washed into fissures in South Wales. And those fissure fills are taken, are prepped, and the individual isolated teeth are collected and similar ones. And when we've found individual skulls, we can then line up all the teeth. Oh, yes, okay, this is that. And we sort it all out. Very nice. Happy days. So what can we tell? Well, actually, teeth tell us a huge amount. Okay. What we've got here is the tooth root. And we've got all of the material around the root that sticks it into your jaw. Okay, teeth of mammals are different from teeth of reptiles. Teeth of mammals are, they have roots and they're stuck to the periodontal ligament. Teeth of reptiles tend just to be sitting in a socket. They're not as informative. They're not as exciting necessarily. Although you can still get the microwear data and all that sort of thing, so you can see what they're eating. You get more information out of the teeth of mammals. If we look at this very outside layer here, let me just go back and show you that one. Okay, so there's material all around here. What you can see is there are these lines. There's a white line and a dark line. And depending on which one, depending on how you've scanned, you either have a white or dark or light. But, you can tell from the thickness of what you have. Here's a Morganeucodon tooth. Here's a Cuneiform tooth. And this is what we're scanning. This is what we're looking at. This is the tooth root, and highlighted in green, this is this outer layer here of something called cementum. And cementum is a fascinating and brilliant material because it sticks your tooth, cements your tooth into your jaw and bonds it to the periodontal ligament. Stops it falling out of your head. What's really cool about it is that it lays down an increment, a dark and light increment every single year. And we can tell that again from looking at teeth of captive animals where we've looked at their teeth post-mortem. We can see this in humans. We can see it. There is this increment every single year, and you get a thick increment and a thin increment, which corresponds to sort of seasonality. One of the really exciting things is you can start to tell things about life history. You can start to tell the difference between males and females, because males have, sorry guys, I'm going to say something which is but it's true, so go with me on this. Males tend to have easier lives, and so their increments are all nice and clear cut, and females have these episodes where they are sucking calcium out of their bodies as part of the hormonal cycle, and their lives are just generally males sit around and have a much easier time of it. So we can see really clearly a male's increments. The increments of females tend to lens more and merge, so you have to look at more areas around a tooth to be able to find the actual increments. But so you can tell between males and females. We have some evidence that that is true. We have, unfortunately, we had one male tooth to be able to show this, but we do see this in teeth. We have very clear increments and we have lensed increments. The clear ones, we are now strongly suspicious that these are male female. The other thing you can see the difference is the difference between females that have just had the monthly cycle But the difference between, or you know, sort of the estrus cycle, I should say, it's not necessarily monthly, and females who have had pregnancy events, because this is where it really gets exciting. If you're going to build a skeleton for your offspring, you've got to get calcium from somewhere, and you can't necessarily get it all from your food. A lot of the calcium will be sucked out of your bones. And going back to the birds for a moment, one of the things we'll also be able to tell if we get to ever scan these fossil birds is birds, also females, make eggs, which are calcium, have a calcium-based shell, and they get the calcium from their bones as well. So you can tell the difference between males and females because the way that the calcium is removed from the bones. And you can also see the calcium being removed from the teeth when you've got these events of pregnancy. So this is remarkable. But we're talking about these two little animals, Morganucodon and Cuniotherium, that branch off down here. Here we go. Where have we gone? In the Jurassic, Morganucodon coming off over here, Cuniotherium coming off over here. These are all the exciting events along the way to becoming crown mammals. We've got monotremes over here, we've got marsupials, and we've got placentals here. This is our branch. These don't make it through. These are on the stem. These are up in the crown, if you like. So there we go. What is going on with these things? What are we looking at? We're looking at Morganucodon and Cuniotherium, and they are small. These are small mammals that are running around in the earliest history of the Mammalia forms. And this is where the funny thing comes in, because I've just told you that we can take cementum, we can image cementum in extant animals, and we can count the number of increments. And so we can age that tooth post replacement, and we can look and see how old that animal is. We can take a shrew, a little tiny mammal, and we can count the increments, and we would probably expect to see 2, 3, 4 would be a pretty old shrew. I don't think there's going to be many 4-year-old shrews. They live fast, they die young. Hang on a second, what have we got here? Here's Morganucodon. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14 increments. Wow. Cuniotherium, 1, 2, 3, 4, 5, 6, 7, 8, 9. That is a lot of increments for a little tiny shrew-sized mammal. So what does that tell us? Here is, okay, so our paper, which we spent ages writing and went through lots of revisions, absolutely brilliant, wonderful PhD student from Bristol, Melissa, and she one of the things she does on the side is make these beautiful infographics, and I'll show you a little bit more in a moment. So here is this is part from her infographic here. This is what went out with the press when our paper was published. And here is our, I'll come back to that in just a moment, but here is our age estimates. We've got Cuniotherium in purple. We've got Morganucodon in the sort of sandy brown. And here we have 8 and 9 years old for Cuniotherium. Here we've got 10 years old for Morganucodon. 14 is an outlier, okay? And you might say, well, hang on a second. I mean, hamsters probably live for 3-4 years in the wild. But, I had a pet hamster and it lived for 10. Yeah, you had a coddled hamster who wanted for nothing, who was given all of its food. Yes, these were not living, these weren't domesticated. These were living in the earliest Jurassic, okay? These were wild. So these are real years, okay? This is real ages, 10, 11, 12, 14 years. That is a huge aspect, a huge discrepancy between what we know about wild living mammals of that size today and wild living mammals 200 million years ago. We had a look as well at long bones, and we looked to see about the size of the femoral foramina, which are where the blood vessels flow, and it gives us an idea of how active these animals were. And what we can see, here's Morganucodon here, okay, we've got reptiles here, we've got mammals here, and you can see that the size of the blood vessels related to body mass. We have much higher, much larger blood vessels in mammals compared with the blood vessels in reptiles, and Morganeucodon sits somewhere in between. We can take a look and we can see lifespan against body mass. And again, reptiles have a very long lifespan compared with mammals for the same body mass. And Morganeucodon and Cunitherium are sitting somewhere in between. Here is the entire, so our paper is 10 pages long, and Melissa managed to reduce the whole thing to this beautiful infographic, which was a page. It's really nice. I've got it stuck up at work. It looks fantastic. What we can see here is we've got animals which are somewhere between their reptile ancestors and their mammalian descendants. So what we've got here is we've got animals which are showing exactly what we would expect. Mammals didn't pop into being fully formed as mammals, but we've got this beautiful mosaic of character acquisition over time. We've got bones which are showing blood vessels which are demonstrating a level of activity somewhere between mammals living and reptiles that are living, somewhere in between. We've got an example here where we've got lifespan, which is more like a lizard of the same size than a mammal of the same size. So we've got this hangover from the reptilian ancestry into the very first mammals. And so all of this is shown from using three-dimensional modeling, three-dimensional imaging and visualization, and why we simply wouldn't have got any of this really from just relying on 2D. And that's it. I will be very, very happy to take any questions, and thank you ever so much for listening. I should say one last thing. Actually, there are lots of people who've been involved in this. Katie Williams at Southampton, Chris Barker at Southampton, Ellis, who's now at Bristol, Pam Gill, who's at Bristol but lives on the island, Philip Schneider, who's in Southampton, Ian Korf, and Melissa. I should have changed that. She's now Dr. Melissa. She got her PhD. She did her Viva and passed last week. So we have these wonderful people. And then all of the people at the bottom here, Katie Rankin, Rich Borden, Mark Mavrocordato, and Orestes Katsiminas. These are the people who do all the imaging and help. So I get to sit here and talk about my PhD students' work and how brilliant they are. And then I get to also have to thank these brilliant people who help us make it all work and come together in the first place. So yeah, it is a joint effort, and I'm very lucky to be able to be the one who sits here and tells you all about it. If you'll just two last things, and then I'm happy to just let you have a disappear and have whatever you need to do, get a cup of coffee or something, and then I'll take questions. Do have a look at the Dinosaur on Your Windowsill, which is a Facebook group, as William alluded to. At the moment, we've got 6 continents, 43 countries, and as of this morning, we went over 3,900 people. 3,900 sounds better than 3,900. 3,900 members. We're still trying to find someone who works for the British Antarctic Survey so we can turn Antarctica red. And I'd very much like to find someone who's on the International Space Station, although I do have, I'm in touch with someone who is from the Netherlands, who has a brother-in-law who is a member of the European Space Agency. So I'm very hopeful that he'll be jetting into space at some point soon. Also check out SOTSEF, which is the Southampton Science and Engineering Festival, and come and listen to Darwin's Armchair, where Sarah, Darwin and I are talking to various people, or just each other, about Darwin and evolution, and having a jolly good time. And I'll be quiet now.
Thank you, Neil. We won't let you be quiet for very long, that's for sure. Okay, let's get some questions into the chat for the break, because I know Neil's very enthusiastic about responding to your questions and having a good general discussion about it all. Before that, a quick heads up on our next talk. On Monday, the 5th of April at 7:30 p.m, Andrew Sieber returns with Immunity Part 2, Cells of the Immune System. That was one of our Immunity Part 1 was one of our first online talks, and I think we had a record attendance for that one. So Andrew's great explainer of all that stuff, and it's about as topical a subject as you get, I guess, at the moment. Anyhow, we will start the Q&A in a few minutes, but in the meantime, feel free to turn on your microphones and cameras and chat amongst yourselves. If you click on gallery view, you'll see more participants as well. And please get some questions into the chat for Neil to respond to when we kick off in a few minutes. Thanks.
Thank you.
I was thinking about what you were saying about difference between professionals and amateurs, Neil. I was thinking, yeah, you fall into that category of people who you're both.
I absolutely. I don't like the word amateur. It's used. I don't know. I don't get to spend as much time digging in the ground as I would like to. But I'm not finding. I'm absolutely reliant on people finding things and handing stuff in, and I think it's very important that we acknowledge their part in all of this.
Yeah,
it's just as important. It's just as important. Sorry, please.
I was going to say I could completely imagine you doing this. Regardless of whether it was your job.
I see what you mean. Yeah, I have to be honest. There are times when it's lucky that Claire is Claire and she tolerates and actually quite enjoys. I mean, we'll be outside as much as we can be. As soon as we're allowed out, we'll be out and doing all sorts of things. If nothing else, we've been promising nieces and nephews that we'll take them over to look for dinosaur bones for the last I don't know how long. So I think we kind of have to get on with that. But no, I mean, Southampton water is amazing. People, you don't look at it, but if you're walking along, we go out, and when we look, we come back with flint sea urchins and all sorts. There's loads out there, beautiful. Some of them are better preserved than others, but they're all there. It's just a case of looking. Yeah. So I mean, yeah, absolutely. I would be doing this if I was paid for it or not. I have the added bonus that I am paid for it, so it's nice.
Yeah.
But I mean, it's one of those things. So I'm a lecturer now, which means I don't have. It's funny when you're a PhD student, you want to be a postdoc. When you're a postdoc, you want to get to be a lecturer. And when it's like all of these things, as you get more senior, you start doing less of what it is that you're really passionate about. I'm lucky because I really love the teaching side of things as well, and so I mean, so that's what I do. I mean, I get to do teaching and balance the research, but the research is supervising really what other people are off out and doing. So it's you're not at the cutting, you're not at the rock face, for want of a better term. But no, I'm very lucky. I love what I do. And I get to work with these really good students. I mean, the students I'm very fortunate. I really am. They're awesome.
So wasn't John LePage one of yours?
No, he was. No, well, oh goodness, that would have been good if he was. I came into contact with John tangentially. No, John was at Warwick, and I'm rather hoping that maybe one day I'll be able to tempt him back. He's a really good. He knows his bones, extant bones, living bones. I think he'd be really good to come and to get involved again and link the present with the past, because the past can tell us about the present and help us make predictions about the future, but we can use the present to try and understand the past better as well. And I think that the idea that you can be one or the other is it's not possible now. I think you have to work together, and you have to bring different ideas and different opinions, and that's also important. It's not just different disciplines, but actually having a really good mix of different people, male, female, every wonderful type of person under the sun, coming and bringing different ideas and different ways of thinking about things, and we'll do better. We'll do better science.
Indeed. And John, of course, has the advantage as well as having a proper evolutionary biologist's beard.
Yeah, well, I'm working on it. But again, it's the 21st century, and I don't want anyone to feel that they can't have a beard. Okay, there's all kinds of medical advances now, which means that actually everyone who wants a beard can have one.
Oh, this is true.
Yes, I don't want my 11-year-old niece to think that she's ruled out of becoming a paleontologist because she doesn't have a beard.
Well, absolutely. All for equality of opportunity.
Absolutely.
Well, I can't see many empty chairs, so maybe that people just haven't got back yet, or but I think we're pretty much there. So I'm going to resume the record and have a look at the chat. So, question from Nigel Martin. Nigel, do you want to unmute and ask your question?
You use the word embryo, but you use it in a way which made me think you didn't mean what I think of as an embryo. What do you mean by embryo?
Okay. So I think of an embryo as an unborn animal, is what you've written in the chat, and that is exactly what I mean. So these little eggs, they are the eggs. So an embryo is all of the stages that are happening inside the egg. So before growth, really, because it's not feeding, it's relying on the resources that it has that have been packed into the egg by the mother. And the embryos that I was talking about initially, the sort of the fossil ones, from the earliest Cambrian, sort of middle Cambrian, about 525 million years ago, are the eggs and cleavage stages and late stage pre-hatched larvae. So they're still embryos because they're still inside an egg membrane. They haven't started feeding yet, of worms. And there are also some embryos of animals which are jellyfish-like organisms. We've got something which may or may not be closely related to insects. The way that it sort of adds its cells and develops is suggestive that it's a sort of arthropod-like, but we're not 100% certain. So yeah, these are absolutely things which were developing with inside eggs, and we have quite nice developmental series of 1 or 2 taxa from the Cambrian. So that's not incredibly informative when you've got 1 or 2 taxa. But if we find more embryos and of different things, then we can start to ask questions about the order of character acquisition and the way that animals differentiated from one another. Because if you look at development, that gives you a good understanding of evolutionary relationships. If things are developing in a very similar way, then they are more likely to be closely related than if organisms are developing two sort of parallel pathways and then maybe come back and converge on a form. But if you've got similar developmental pathways that are shared, that's really showing evolutionary relationships. And Darwin talked about the importance of development right back in the Origin in 1859.
Thanks.
It's okay. Thank you for your question.
And Verity, do you want to ask your question?
Hi, Neil.
Hello.
I was just wondering if you got a bone from the reptile mammal, one in your last slide.
Yep.
And you did some CT scans on it, looking at the blood vessels. Can you actually tell if it's warm-blooded or cold-blooded, depending on reptile or mammal, from the CT scans?
Well, what we've done is we've got the bones, the long bones from these Morganucodon and Cuneiform, and we've looked and we've seen the size, the relative size of the blood vessels in relation to the length of the bone, etc., and the size of the of the cavity inside. What we've then done is we've looked at reptiles and mammals, and we've seen that the mammal one, the true living mammals, have a much larger size blood vessels compared with living reptiles, and the Morganucodon and Cuneiform, these stem mammals, are somewhere in between. When we've then related that to the teeth as well, showing that they are very long-lived, we've shown that they weren't as energetic. They have a lifespan which is equivalent to a lizard of the same size, reptile of the same size, as opposed to a mouse of the same sort of body mass. And so that gives us an idea that they're on their way to being fully mammalian and fully warm-blooded, but they haven't quite got there yet. Does that answer your question?
Yeah, it does. Thank you.
That's my pleasure.
Really interesting.
Thank you.
Right.
Hello.
Hello.
Am I coming through?
You are?
Oh,
Go ahead, David. It's okay.
how recently can you attribute intelligence to these creatures?
Well, I suppose it. Sorry, I interrupted.
No, I was just saying, or don't you attempt to?
I don't know that we can attribute intelligence to any living mammals. Well, no, that's not probably very fair. I don't think we can attribute necessarily intelligence to human beings, but there are other animals which are obviously more intelligent than others. The brains are much larger. Okay, the brains of these mammals are much, much larger than the brains of equivalent reptiles of the same body mass. So they are, again, moving in that direction. Exactly what we can work out with intelligence. There's a chap by the name of Tom Land who did his master's, his MRes with us last year, and he was looking at primates and trying to work out intelligence and how that was related to vision and to sort of food choices and all sorts of things. So that we're looking a lot more closely to us rather than sort of and groups that are more closely related to humans, because what we mean by intelligence, I suppose it's fairly it is something which is personal to us. We know what intelligence is, and so equating it and recognizing what we might understand from fossils has to come, I think, from things that we would recognize as being intelligent today. That's not a very good answer. I apologize.
At that period of time, I would have thought intelligence would be sort of judged upon their adaptability.
Well, I think that's exactly it. I mean, I think the increase in brain size was absolutely related to these organisms being able to take advantage of niches, take advantage of opportunities that were. I mean, the dinosaurs and the reptiles were dominant. They were, and so there would have been lots of competition amongst these stem mammals to be able to. Dominance isn't the right word. We've got dinosaurs coming to prominence throughout the Jurassic, all the way up to 66 million years ago. There was no room for mammals to become dominant, but they've got to be able to hang on in there in some ways. And I think this mosaic of characters, this becoming warm-blooded, and so becoming much more active at night and being able to take advantage of different niches, different opportunities, meant that they were almost pre-adapting themselves, if you like, to when the extinction event happened 66 million years ago, and niches then opened up. And I think certainly having a larger brain at these points would have been extremely helpful, so that they could take advantage of opportunities. Does that answer your question?
Yes, thank you.
Thank you. No, thank you.
Talking of niches, I was reading an article quite recently, don't think it was one of yours, but it was arguing that in the Jurassic, there was a lack of sort of medium to large sized animals.
Oh,
Or species, and they were positing that it was juvenile theropods that filled a particular niche, and therefore you didn't get animals that were that size at maturity because the theropods were so successful. Is that something that sort of you go on with, or?
I think that's really interesting, and okay. So the paper was out last week, and I will be honest, I've been finishing off my marking and doing all sorts of other things. But it's at the top of my list of stuff to read. It is a really interesting hypothesis. We have small theropods and we have large theropods, and there isn't anything in the middle. And it's been. I've just seen that Chris Barker is actually on in the chat, so maybe Chris would like to come in and put an opinion as well. Chris being my PhD student, assuming it's the same Chris Barker, it's the sort of question that he might ask. So yes, I think it's a really interesting hypothesis, and it does sort of explain the missing dinosaurs. I quite like it. I want to go into more details and actually go through exactly what they've been looking at, but I think it makes sense.
Oh, next time, Chris, do you want to come in on that? And you put your own question in the chat as well, I see.
Hi, I've not actually read the paper either. Yeah, the hypothesis seems pretty sound. Initial brief article I read on it, and yeah, we are expecting to be to have a really high contingent of really sort of small to mid-sized theropods, and this applies to dinosaurs in general, because obviously you're going to have a bias towards the younger individuals of any population, because obviously dinosaurs were producing a lot of babies relatively often, and very few of those were actually getting towards the larger sizes. They were getting eaten, they were dying young. So it does make, it makes sense to me. But yeah, I would have to read the paper to have a more informed comment.
Sorry for dropping you in on that one, Chris. Apologies.
Do you want to ask the question that you put in the chat at that point, Chris?
Yeah, sure. My question. Yeah, Neil, I was just wondering what you thought about the new hypothesis that posits dinosaurs were lagoon denizens and incapable of terrestrial locomotion, as published in Brian Ford's recent book, Too Big to Walk.
Oh, I remember this one, Chris. Thank you very much, sir. Okay, so Brian Ford has put forward an interesting hypothesis that dinosaurs were enormous and too large and simply wouldn't have been terrestrial, and actually would have been walking around in lagoonal habitats, walking across lakes, living. That's a nice idea if it had any major support, and that there isn't any really. Dinosaurs are found in all habitats. They're not just found in lagoonal, to keep using Chris's word. They're found in it. It's a nice idea, but there isn't the evidence to support it. And actually, when we start to look at the biomechanics, when we start to look at the way that we believe dinosaurs are put together, there's actually no requirement for them to be living in water as opposed to living on the land quite happily. And they did it for 150 million years with no problems whatsoever. It's an interesting hypothesis, and it's one that I happen to know that Chris doesn't agree with either. So.
Neil has also said that he hopes to be able to give us an update perhaps later this year on some new research that hasn't been published yet. So perhaps by that time you'll have had a chance to read the paper about the teenage tyrannosaurs as well.
Well, I think Chris has just has definitely booked himself in an opportunity to read that. I shall definitely be, suggesting he does.
If I can cut out now, they're just talking questions. It's quite interesting.
Background noise.
Mm-hmm.
think we killed that, right. Don, sorry, we've taken a while to get around to your question.
Hi, William. This is a fairly simple question, I think, really, but it would seem to me that you've talked about a meeting of minds, different academic minds,
Yes.
bringing about your modern hypotheses and modern methods of looking at your paleontology. And then I go back to a question, of those, who perhaps was the prime mover? Was it the imaging experts who were looking for an application for their academic discipline so that they could continue to get more funding, or was it paleontologists looking for some way of getting over the hurdle of can't see inside a piece of rock?
Yes, and I think both of those to an extent. However, we can go back to the sort of the early 30s, 40s of the 20th century, and we can look at people like Jarvik Stensio in Sweden, who had a huge sort of 5 meter long, 3-dimensionally preserved chunks. Well, sorry, the chunks of 3-dimensionally preserved fish where the individuals would have been several meters in length. Of an animal called Eusthenopteron, and Eusthenopteron is a lobefin fish, ancestral to living tetrapod vertebrates. Us. It comes off the bottom. It's closely related to us by him. And what Stensio did was sort of a bit halfway between what we were doing with the CT scanning, although our CT scanning isn't destructive. It's brilliant, really clever. What they did was they took a fossil and they ground off a millimeter, and then they drew that bit, and then they ground off another millimeter and they drew that bit, and then they ground off a millimeter and drew that bit, and so on and so on and so on, and built up a tomogram destructively, but looking at the internal structures. Well, the technology didn't exist then to be able to do high resolution CT scanning. In fact, if you stick something into the scanner which is a meter in length and great big solid lump of rock, you aren't going to get great resolution either, because you have to increase the energy to punch your X-rays through, and higher the energy X-ray of the X-rays, the lower resolution that you can get coming out of them. And the contrast goes as well. There's all sorts of other issues. But the technology improved. A chap at the Swiss Light Source it was quite funny. Phil Donoghue was working with Dong Xiping, who had the fossil embryos. And the chap in Switzerland contacted Phil and said, would you like to have a look and see what's what? So it is a meeting of minds. It's brilliant. Phil did some initial work, and then I was very lucky to then come in quite near the bottom of the near lower stories of the tower, if you will, and yeah, had a lovely time scanning all sorts of material. The applications aren't just to looking at sort of millimeters diameter embryos. You can scan charcoalified flowers from the sort of Cretaceous. So the earliest flowers, you can go back to the Carboniferous, and you can scan little sort of coniferous, sort of gymnosperm cones and what have you, little tiny structures, which are beautifully preserved as charcoal fossils in 3 dimensions. And as soon as you've got that, you can get wonderful biology coming out of your rock samples. And it's all of these things coming together. The appliance of the physics along and making the scanning better, the computer scientists bringing about the algorithms for reconstructing all of because you've got to remember as well, okay, this isn't just an X-ray where you've got your hand, boomf, okay, you rotate everything ever so slowly, and as you do that, you get slightly different images, and then you've got to put them all back. Now, I don't have the mathematical or computer science understanding to do that, so I can bring the fossils and have a think about the evolution, and some genius with a computer can make these brilliant algorithms that allow you to reconstruct all of these little tiny points into 3 dimensions, and to give you, rather than a pixel, which is your 2-dimensional square on your television screen or your computer monitor, you can make it a voxel, which has got the Z dimension as well. And then you can start to move between slices, and you can trace structures through, exactly the way as Stengio and Jarvik did with Euthanopteron and paper. Yeah, and you've got your internal structures, and you can separate geology from biology.
I've never heard of a voxel before. I shall hang on to that concept. It's a
Well, when you start to find out a lot of things that you didn't know before when you start getting into CT scanning and imaging.
Yes.
When you look inside your body for the bits that have just gone rotten, and you get a chance to look at the CT scan, you begin to realise.
Well, because then the next thing that you can do, so there was the CT scanning, and it gives you the structures. One of the exciting things that I did was I took eggs and embryos of marine worms and starfish, things which were ethically non-problematic, although I still went through all of the ethics approvals and everything to work with them, and let them go through the stages of decay. So we're talking about cellular structures here, little eggs of Pomatosauros, the little worm that's on the bottom. You find seashells in the intertidal. You've got this little white tube. Well, there's a worm that lives inside of that, and looking to see what happens when an egg decays, because a fossil isn't perfect. A fossil has died. It will have gone through some processes of decay, and so if we can start to understand what those processes look like, what those structures look like, then we can go back to our fossils, and we can start to go as well when we're looking at our beautiful 3-dimensional images from the CT scanner, saying that's geology, that's biology, that's what happens, that is a decayed gut or whatever we're looking at. But you can start to tease these things apart, and it can be smelly, but it's fun, and it's a really good way of the fossil record becoming something which is really informative about the way that characters have been acquired in evolution of different groups. That's the aim. That's the hope, is that we'll have enough of these different embryos, we'll understand them as well as we can, and we'll be able to tease apart what the signal is in character acquisition. Because, for example, you've got arthropods, and this is important for understanding human development as well. But you've got 2 model organisms. You've got fruit flies, Drosophila, and you've got nematode worms. Now, they're really closely related to each other. They're not quite the cousins on the tree. There is a couple of steps in between, but nematodes are a little roundworm with very little. I love nematodes; they're brilliant, but they are not the most exciting looking organism. It's a worm, and it has every single C. elegans we've looked at has 959 cells in its body. Well, a fruit fly has limbs and wings and complex sense organs and all sorts of other structures. C. elegans, the nematode worm, does all sorts of things, and its pathways are the same as ours. So if you put alcohol on its little dish, it will behave neurologically in the very similar way to the way that we behave neurologically when we have too much alcohol. So you can model all of these things, but actually the way that characters are added in evolution, you've got these 2 really closely related groups. This is all happening in the Cambrian or just before. And we don't know why one branch has gone to form a little worm, which is quite simple, and another one has gone to form a very complex, multi-limbed, multi-sensory, lots of different sense organs. And so being able to find these physical fossils, which put a pin in it, if you will, of when characters were being acquired. That's really crucial. That's really important. And that's what we hope to be able to do. And the more places we explore, rocks of the right age, the more fossil embryos we're finding. And we're not finding hundreds and thousands of different taxa, but we are finding exciting things all the time. So we keep looking.
Thank you. And Sam, sorry, you've had quite a long wait for your question. Do you want to ask it now?
All right, it's been interesting to listen.
Hello.
I'm just wondering, hello. You mentioned about being able in the future to be able to sex the Archaeopteryx fossils. For a while, it's been possible to, age dinosaurs and early birds and all that from growth rings, and then possibly be able to sex them due to the presence of medullary bone.
Yep.
Would this be a possibility with Archaeopteryx?
Yes. Long and the short of it, yes, absolutely. so the medullary bone is where it's going back to sort of eggshells and all this sort of structure. You can tell the difference between female dinosaurs and male dinosaurs and female birds and what have you because of the presence of medullary bone. It's where the bone is is modified when females are drawing calcium out to make eggshells. It's one of the reasons why battery chickens are such a terrible way of farming chickens, because they're producing eggs so quickly that their legs that the calcium comes out of their legs, as it were, and so they end up with bendy bones because the bones are so under calcified. So that's just going to explain what I'm talking about getting the calcium from them. So yeah, hopefully at some point we'll be able to tell the difference between males and females. Another group of birds which are very exciting, about 125 million years ago, so much younger, 25 million years younger than Archaeopteryx, and with more modern bird, more derived bird characters, is a group called the Confucius ornithids, and they may or may not be showing sexual dimorphism, as in the males may have long tail streamers and the females not. Well, what we want to do is actually look and see if that's the case, and by using CT scanning, eventually when the technology is able for us to scan at the right resolutions and see, do we see medullary bone in these ones with long tail streamers? Because if they've got medullary bone and they've got long tail streamers, then they're not males. Equally with Archaeopteryx, what we hope to be able to see is, are they mature? Because there are three or four species. We've got Archaeopteryx lithographica, we've got Archaeopteryx simensi, we've got Archaeopteryx. Oh, it's got out of my head. That's terrible. We've got to, it'll come to me in a moment. It's always the time it gets embarrassing. I don't know. Are they different species, or are they different growth stages? If that's a sort of it's not going to be a development. It's not going to be a developmental series, but maybe it is. Between Archaeopteryx grandis, there we go, the big one, the Solnhofen specimen, lithographica simensi, and are they different species? And we hopefully would tell that, potentially answer that question by looking to see if they're mature. If they are if there are juveniles and mature ones mixed up, we would lose the juveniles, because we don't use juvenile characters in most cases to identify a new species. We tend 99% of the time to use fixed adult characters for our systematics and taxonomy. Not always, because sometimes you have to work with what you've got, and fossils are a case of it isn't perfect, the record isn't perfect, so we do have to have a think about, on balance, try and have a look and see what we've got. But the hope is, sorry, I should answer your question really, the hope is that we should be able to, when the technology works, to have a look at things like Archaeopteryx, because again, the possibility of us being allowed, ham-fisted as we are, to get hold of these absolute wonderful fossils, which so beautifully outline evolution and so beautifully suggest. I mean, these are they're some of the most important, beautiful fossils in the world, and museums are not just going to let people go off and CT scan them. But if we can do it in other things, we can start to get an understanding. There's 100 or so Mesozoic birds that have been given names. Are they real? As in, do they all deserve their own species name? Are they all different genera? Are they? So these are the sort of questions that we want to answer. And it's not for us to be saying, you know, being killjoys and saying, oh no, we've got many fewer. It's about being accurate and about what we're talking about. And if we've got more, then that's the other possibility as well. Because if they're all mature and they're all different, then they're all species, with lots of caveats in and about how we do. Does that answer your question?
Yeah, I was wondering why we hadn't done it already, but if it's the limitations of the technology.
The limitations of technology, because the other problem as well, okay, so the perfect thing to scan is a long, tall cylinder, because then the X-rays pass through it in every direction through the same amount of material. When you have a fossil like Archaeopteryx, which is on a slab, you've got X-rays passing through in one direction through, you know, millimeters of rock. In the other direction, you know, 30 centimeters. And so the X-rays coming out the other side of a few millimeters are much more intense than the X-rays passing through the long axis. And when that happens, you lose contrast. You can't build beautiful stacks of images that you can then analyze. So we're working on different ways of imaging and doing all this sort of stuff all the time. And MUVIS is at the front, really at the cutting edge of doing all of this.
Oh, thank you very much.
Pleasure, thank you.
I got a question on YouTube. Somebody saying, did you say there were no marine dinosaurs?
Oh dear.
Graphic Life.
There's a question. With all of these things, there are exceptions that prove the rule. Dinosaurs are terrestrial organisms. Dinosaurs are not flying organisms. But I've just told you that birds are dinosaurs and birds fly. So your traditional dinosaurs, Stegosaurus, Brontosaurus, T-Rex, they're all terrestrial. There was a paper in Nature, made the front cover of Nature very exciting, that Spinosaurus aegyptiacus is an aquatic animal and is potentially a pursuit predator. However, I'm not convinced. I think that absolutely so these are Spinosaurs, and so it includes a group that live on the south coast we've found representatives in the south of England. So the Baryonychines. In North Africa, we've got Spinosaurines. So two families of Spinosaurs. Baryonyx and its ilk are sort of about 125 million years old. The Spinosaurines are significantly younger. And Spinosaurus has been described as being aquatic. I think that it might be, but I also think there's a very good paper out last week, a couple of weeks ago, Hone and Holtz, which is Spinosaurs are one of the most incredibly controversial groups as well, and I've managed to get myself involved with Spinosaur research. What was I thinking? But the Hone and Holtz, I think, present a very balanced critique where they show that it might be aquatic, but actually there are other explanations that are equally parsimonious. So long and the short of it is no, dinosaurs on the whole are not aquatic. That doesn't mean to say that some of them might not have had a more a closer relationship with water. And Baryonyx, absolutely, the specimen that was found from smoke jacks in Surrey in about 1986. It's beautiful. It's up on display in the Natural History Museum. It had fish scales found in the sort of the stomach region, which one of the projects we're hoping to get off the ground at some point is looking to see if they're etched by acid to see if they've been eaten. But I mean, it has the Baryonychines and the Spinosaurines have this classic sort of crocodile head morphology. They have teeth which are sort of individual points rather than serrated, like and sort of blade-like, as you find in Tyrannosaurs. So they are almost certainly eating fish, and certainly look like they have an element of their diet is fish. But whether or not they are swimming and active pursuit predators, as was put forward middle of last year, or if there's other explanations, I think the jury is still out a little bit.
And Cleo has asked, how much biological tissue is left in what you were looking at, and how much is replaced with minerals?
Well, okay, so with fossils, pretty much everything there's no, in the material that I'm looking at, all of the biological material has been replaced with minerals. So what we're doing is looking to see how accurate that replacement is and how much of a representation of the biological structures we have, as opposed to being completely replaced and, you know, lots of decay has happened. If lots of decay has happened, then you end up with mineral infilling, and it's a bit of a mess. And that's what we're trying to untangle here. So there is, in almost all fossils, there's next to no biological material. Some long bones of dinosaurs use some, you know, there've been reports of proteins, and that's pretty, I'm pretty happy with those being proteins. There's a lot of, there's some T-Rex protein, collagens coming out of a long bone. I think those are absolutely proteins, and from the T-Rex, I'm quite happy with that. But the vast majority of material, 99.9% plus of fossil material is mineralogically replaced, but some of it replaces and produces a better representation of the biology than others. And this is where this sort of technology can pull things out and start to show us that.
Great, thank you. And that brings us on to one final question, which has been asked on behalf of Claire. What is your favourite fossil and why?
Okay, I have mentioned it before. My favourite fossil is absolutely Archaeopteryx. And it's not because it is the best preserved fossil in the world. It's because of when it was found and what it supported. And as I said earlier on, Darwin wrote in 1859 that it's frustrating that we don't have the fossil record showing us these beautiful transitional forms between the great classes that exist today. And he wrote chapter 10, The Imperfection of the Geological Record, and of the origin. He goes into this great detail. It explains that it should be there, and then just a couple of years after the origin was published, we find the feather, and a couple of years after that, we get this large body, whole body fossil, which shows features which link birds to reptiles, and problem solved. Evolution, tick, no longer controversial, all arguments, and therefore are resolved just 4 years after the publication of the book. And no one has ever questioned evolution since.
Splendid. Well, that brings us to the end of the formal meeting. Many thanks indeed to Neil for a fascinating talk, and to you all for attending.
Thank you.
And we look forward to hearing an update on Neil's research in due course, and we look forward to seeing you all next month.
Thank you very much for having me. Thank you.
Thank you.